Let's move on to the last section of our program, which is Gayane again and Andrew Mould. And we're going to talk about the study wrap-up, analysis, and publication phases. Gayane, will you start and then introduce Andrew?
Absolutely. So yeah, so at this point, in the trial, the teams have to be working seamlessly. So that was the motivation for this presentation. We try to create a blended presentation between me and Andrew to kind of mimic that process that at this point in time, the statistician and the operational managers will be really in full a blended team. And so Andrew Mould is an assistant professor in the Department of Neurology, and he directs the clinical trials within the BIOS Clinical Coordinating Center. So I'll start, as Dan said, and then I'll give it to Andrew. So next slide, please. So again, to emphasize where we are, we're now in the last phases in data analysis and publication, making sure everything is working, and then wrapping up the study, presenting the findings, and then sharing the data. Next slide, please. So the enrollment is almost complete, and now what is where we're starting here. And so again, there are a couple things that we want to highlight here, and then we'll talk in our presentation a little more in detail. So we have a statistical analysis plan that maybe can use some final touches. We have analytic timeline that's been developed based on, again, thinking from the end, when the study data should be presented, and then thinking back how much time it takes to do all these activities and lead us to results and the sharing of the study findings. Last checks on the data completeness and data validation, essentially what Lindsay was talking about, data log plan and timeline, and then manuscript production plan, unblinding, data analysis, and data archiving. So this is essentially the scope of this blended presentation. Next slide, please. So the data completeness and data log, and again, you've heard today a lot about the implementation phase and how we monitor the data, how we look at data completeness, the outcome, data collection. One thing added here that I don't think was mentioned before is that we, in the statistical analysis plan and the protocol, we define the outcome data windows, and then kind of maybe can look at the outcome data collected outside the outcome windows, and then see what that proportion might be. Again, the plan for that should be already established in our plan. The final missingness assessment should be happening, and then attempts made to then complete the data, make sure the data are entered, cleaned within the sites, again, work with the sites, motivate the sites, the strategies that Lindsay was talking about. At some point, again, we have to put in a timeline when the final queries should happen, and then all queries resolved. And again, as I said, we have a timeline for all these processes, including the data log, including the final sign-off by the site and coordinating center teams. Next slide, please. So this is what I mentioned about the SAP. We view SAP as a living document, so there is still a little bit of a time to put final touches to the SAP prior to unblinding. Again, if everything planned well, ideally there is no additional guesswork at this stage. All the statistical analysis are defined clearly in the SAP with all the definitions, the roles. And again, Andrew will talk a little bit more about dual programming, but the roles, the processes, the code that essentially defines the entire workflow from raw data, from the EDC to the analytic data sets. And all of this is defined, tested, and confirmed that everything is working. Now, why do we say that there might be still time to do some changes? In one of our studies, for example, we kind of pre-plan these because some new data might come to light that would affect the study outcome and planning. And what do I mean by this? So again, depending on the disease and the condition you're working on, so this was specifically a trial for Alzheimer's disease, we kind of knew that this is a rapidly developing field. So there might be, by the time the study is coming to the closure, there would be some additional biomarkers that might be relevant to the study. And so what we did is we pre-planned that there would be some additional assessment of the field and which biomarkers might be then again added or replaced and put in a secondary endpoint, tertiary or exploratory. So this was specifically the SAP was written with that in mind, that there would be at some point this assessment and we'll look at it and there'll be selection for that. And of course, allowing for enough time to do that, taking into account the data timeline and the analysis timeline is important. We also might have some additional surprises, but again, this won't be some things that would really dramatically change the SAP. It's just something that would be additional emphasis in our analysis, such as higher than expected missingness, shorter follow-up for some participants for whatever reasons, and again, outcome data that is outside the window in proportion of that relative to the outcome data that is collected per protocol. So this is, again, the last chance to update the SAP prior to finalizing the document. Next slide, please. And so this is the now thinking about the analytic data sets. As I mentioned on the previous slides, is that the whole process need to be tested and confirmed and the roles defined clearly. So this is really the whole process of transforming the raw data into analysis-ready data. Again, key points are documentation and reproducibility. We want to make sure all the steps that are involved are clearly outlined and are auditable and reproducible. The data cleaning rules, any derivation logic, scoring procedure, merging procedure of different tables in the EDC, and so forth. So this is something, again, Josh mentioned in his presentation that is clearly of key importance. And the last section of this slide again talks about specific data analysis model that defines these things for data set and metadata standards. This is required for FDA submissions. And so again, the principle is efficient generation, replication, and review of the analysis, and then traceability between analysis results, programs, and the data. Next slide, please. So at this point, I'm going to hand it over to Andrew, and he'll continue from this section from the operational point of view.
Great, thanks, Gayane. So at this point, as Gayane mentioned, you've sort of revisited and finalized your SAP, all of the specification work, and you're sort of ready to launch into that analysis. But what this doesn't mean is that the statisticians go off, into a room and produce the output and come back when it's ready, right? This is still a very important statistical and operational collaboration that expectations have to be outlined on exactly, what it is going to take to cross that finish line. So the statisticians have that SAP, that is their roadmap. It's going to be well understood, the tables, listings, and figures that need to be generated. From the operational side, you're looking at, funding, budgets. Is there a target journal in mind? Are there requirements for timeliness of submission for those results after the trial concludes for that target to have, peak interest? Are there conference presentation targets where a therapeutic area's annual conference that's well attended is in 4 months' time, and it would be ideal to have your results shared to such an audience? It's also increasingly prevalent to find grant milestones tied to these key outcomes of the analysis being complete, the manuscript being submitted, or even the data repository step that I'll get into in a little bit. Next slide. Okay, so now we're at the manuscript writing stage, and this is something that should be thought about well in advance, as is the theme for this entire symposium. But authors too fall into this category. Whenever possible, this should be decided at the onset. Publication policies can sort of help here. It will outline your key decisions related to how you're going to go about assigning these tasks and having creditability for the team members in the primary and secondary manuscripts. So for example, it almost goes without saying that the majority of the time the lead statistician is part of that author block. But how do we treat statistical programmers that are doing a lot of that legwork into that final analysis, or junior statisticians included as well in the author block? There are guidelines to help you make these decisions. There's 4 criteria that it sort of takes you through, and that's the ICMJE criteria. But this is best to have a conversation well in advance where emotions and sort of the stress of getting everything complete at the end are somewhat removed from those decisions. How the work gets done is obviously going to be individualized to your trial. So things like writing committees or manuscript working groups fit for size, whether that's small or large committee, might allow you to progress in a timely manner. You might also leverage the existing infrastructure that you have set up for the trial operations and have a routine check-in with committees like your executive committee for looking at the output and kind of bouncing the story off of them as an external validation. At the end of the day, writing should ideally begin in advance of the analysis taking place. There's no reason why the introduction, method, statistical approach, discussion, etc., can't be thought of in parallel to the analyses being done. Sticking with the theme of you got to have the goal in mind when you're going through that exercise, and actually putting pen to paper, figuring out that story that you want to tell might highlight a few more areas of analysis that you perhaps didn't probe properly. There's also going to be constraints around table numbers, figures, what can go into a main body versus a supplementary material, the size of those. And these are all going to be stemming from the journals that you're targeting, but would be good to have in mind rather than creating everything that you want and then trying to pare it down. Next slide. So approaching the validity and reproducibility components. So Gayane introduced us briefly. For the longest time, the gold standard has been dual programming. And although this, double programming is that gold standard for validation and advancement, the advancements in technology and the regulatory guidance have shown that it may not be necessary, right? And there is a cost that comes with this. There are timing constraints. There's the very real understanding of how do you deal with discrepancies when the two don't agree, what third party gets inserted to then make that decision, or how are they resolved. When we look in the literature to see kind of guidance from an FDA standpoint on whether this is required, you'll actually find that even though paradigms like the SDTMs and ADAM that Gayane introduced are required for FDA, dual programming is not. So in other words, it is not a regulatory requirement, and you are free and encouraged to use what often is used in other elements of trial implementation and planning, that fit for purpose, risk-based approach that allows your organization to tailor their validation strategies based on the complexity of the study, the level of risk, the available technology, et cetera. The AI is here and now discussion is applicable here. There are AI-driven solutions into how you might be able to validate part or all of your output that your statisticians derive in a more efficient and budget-friendly manner. But whatever the approach is and whatever is implemented, they should all have that same main goal, which is, do the results make sense and are they consistent? Next slide. So you have all your results, you've published your paper, you're at that endpoint, and then there's the very real need to talk about data archiving, data sharing. This is an increased focal point for NIH and all sponsors, sort of alike. The reproducibility component, the altruistic sharing of data for the greater good of the scientific community. And this has worked its way into being a requirement for most grant applications. So you will need to put in your plan as to how exactly you're going to share the data. Fortunately, they too have also increased their focus on standing up these NIH-sponsored repositories to allow for ease of that sharing. So things like BioLink, BioSend, the Brain Imaging Laboratory, Vivli, et cetera. There's also the separate component, which is all clinical trials are required to be registered in clinicaltrials.gov, which in turn creates the need to then circle back and have results uploaded within 12 months of that primary data completion. These tasks are not trivial, and they are often ones that are forgotten about until the last minute when compliance is an issue and funding is constrained. So it is imperative that in thinking through your timelines, your project budgets, et cetera, that you adequately provide the funding and the effort levels needed for statisticians, data managers alike to ensure that these tasks can be completed in the project cycle. There are many data repository types. So if you're wondering, how do I go about deciding which one is correct? There are institutional data repositories, there's specialists, meaning the therapeutic area that you may be studying, or generalist data repositories. And you really need to choose what makes the most sense for the data generated. So on the next slide, which is my last, is sort of a paradigm of how you might go about thinking through this. So if you need to select a data repository, it would behoove you to first look at the sponsor that you're applying to. Do they have a repository that they favor, that they hope that you will use, if funded by them? And if so, it's probably a smart idea to use that designated repository. If that's not the case, or you have reason to go against that, is there a discipline or domain specific data type repository where you might use that to align it with the therapeutic area being studied? If that doesn't exist, maybe it's rare disease and there isn't, a true database for that area, there are generalist repositories. So kind of agnostic, good for any clinical trial. And there's always the option in that whole fit for purpose where if you have a small study, for example, and your data sets are under 2 gigs, they can actually just be included as supplementary material in PubMed Central with that publication component, and you may not need a separate external repository. Whatever, again, the choice is, recognize that there are usually pre-described formats and an understanding that needs to be had as to what that data needs to look like in order for that to be accepted and built into that repository. And that should not be looked at and planned for, 2 weeks before you need to submit it. But it should be thought of at the very beginning so that that everything that Gayane and Josh and Nicole and Lindsay have been talking about, that this too is planned for at the beginning so that you're seamlessly integrated with the end goal in mind. And with that, I'll turn it back over to Dan and we can jump into the Q&A. Thank you.
Okay, great. Also a wonderful presentation.